Assalamualaikum. My name is Muhammad Najib bin Kasri. My inventory number is S60320 under Dr. Muhammad Hafiz bin Bukharuddin. I will talk about progression and feeding ecology of blacktip reef shark, Cocorhinus melanopterus in the Bidong Island and Redang Island. Introduction. The blacktip reef shark, Cocorhinus melanopterus, is a species or recurrent shark in the family Carinahidae, which can be easily identified by the prominent black tip on its fin, especially on the first dorsal fin and its caudal fin. It differs from the nearest relative blacktip shark, Carcharinus limatus. This is the blacktip reef shark taxonomy. The blacktip reef shark is among the most abundant sharks inhabit the tropical coral reef of the Indian and Pacific Ocean. This species prefers shallow and inshore waters. Research background. A population is a subgroup of individuals from one species that live in a specific geographic area and interbreed in sexually reproducing species. Sexual segregation has important conservation implications for marine population management because it provides information on the spatial dynamic of male and female distribution, abundance, and thus the differential anthropogenic exploitation of the sex through spatial focused fishing pressure. Animal movement and habitat selection are impacted by a variety of biotic and abiotic factors and ultimately determine how effective an individual is at finding food, possibly mate, and avoiding predators. The species feeding ecology is intricately linked to its population dynamic and is contribute to our understanding of topics like resource partitioning, habitat preference, prey selection, predation, evolution, risk effects, competition and trophic ecology, and energy transfer within and between ecosystems. Dissection and investigation of stomach content is a typical method for studying fish diet, feeding ecology, and eating preference. However, various other procedures, including non-linear method of obtaining gut content, stable isotope analysis, direct observation, and fatty acid analysis, have recently been applied. Justification. The black-tipped reef shark is listed as vulnerable on the IUCN list. The measure, especially in Terengganu, there are not so many studies done on the population and feeding ecology of black-tipped reef shark, Cocorhinus melanopterus. The number of black-tipped reef shark keep decreasing because of anthropogenic like overfishing by commercial fisheries for their meat, liver oil, and fin, habitat destruction, and many more. Black-tipped reef shark act as an apex predator in the coral reef ecosystem. While anthropogenic disturbance is increasing, the knowledge of their life history variable and their intraspecific variability is lacking. Objective for this study is to compare the number of black-tipped reef sharks in Bidong Island and Redang Island. Next is to determine the diet of black-tipped reef shark in both islands Bidong and Redang. Next is to determine if the black-tipped reef shark Cocorhinus melanopterus in Bidong Island has a connection with the black-tipped reef shark Cocorhinus melanopterus in Redang Island. This is the sampling site, Bidong Island and Redang Island. It was about 15 kilometers from each other. This is material that will be used during this study. Fishing rod with the bubbleless hook, tape, stable isotope, biopsy sampler, freezer, oven, mortar and pestle, analytic balance, carbon nitrogen analyzer, camera, weight scale, ethanol, phenol chloroform, polymerase chain reaction, megalcy, and tag. For the survey method, first, fish will be caught using the fishing rod that attached with the bubbleless hook and brought alongside the boat. Then the shark will be restrained and turned on its back. Then the shark will be measured, set, picture, tag, and released. After 2 months, the shark will be caged again. For stable isotope method, fish will be caught using the fishing rod that attached with the bubbleless hook and brought alongside the boat. Then the shark will be restrained and turned on its back. A small incursion in the flank of the shark will be made and using a biopsy sampler to remove a small piece of special white muscle tissue. Then the shark will be measured, set, picture, and will be released. Then the sample will be frozen for the preservation. Next, the sample will be dried in a 16 Celsius drying oven for at least 48 hours. Then the sample will be ground into a fine powder and weighed out into micro sampling dish. Then the carbon nitrogen analyzer will be used with the generalized linear model. Lastly, for the phylogenetic tree method, the sample will be preserved in the ethanol. Next, DNA extraction will be done using the standard phenol chlorophore extraction. Then PCR will be performed for the amplify. Next, the data will be analyzed using Megaseq software. And this is the Gantt chart. As you can see, the black one is the had done and the yellow one is will be done. For the expected outcome, the number of the blacktip reef shark at the Redang Island is more than at Bidong Island because Redang Island is bigger than Bidong. Shark at Bidong Island and Redang Island has a connection with each other because they may migrate for better food, habitat, and meat. The shark that will be captured for the stable isotope analysis survive. Lastly, this is the references that have been used during this study.